So hello everyone, today I will be carrying out the first Form 4 project of biology subject. Before that, my name is Alistair McRyan and I am studying at Sekolah Menengah Sains Kuching Utara and currently in the class of 4 Newton 2. So basically, today I will be conducting an experiment related to subtopic 3.4, which is from chapter 3 biology form 4. So this experiment is focusing on the concept of movement of substances across plasma membrane. So what is plasma membrane actually? Plasma membrane is an organelle found in both plant and animal cell, which is to control the movement of substances in and out of the cell. So by doing this experiment, we will get to know whether the solution we will use later on is hypertonic, hypotonic, or isotonic to the main goal that we will use in the experiment. So without further ado, let's begin.
Precaution: wash the spatula and the glass every time after use it in each solution to avoid the mixing of substances. Precaution: use warm water so that the solute easily dissolve. Precaution: make sure the amount of water used is the same to get better results. Wash the spoon before use for other substance to avoid mixing of substances. Precaution: stir gradually so that the mango mixed equally to the substance used.
One week later.
So hello everyone, so today I will be sharing the result of the experiment that I have carried out 7 days ago. So in front of me here are the 3 containers containing sugar solution, salt solution and vinegar. So without wasting time, let us take a look at the first container which contained sugar solution. But before that, so we can see that here the first container have excessive amount of water, whereby the amount of water is increasing compared to the day that we do the experiment. That's why I am using this bowl so that it can fill the excessive water contained in the first container. So we can see here the mango or the plant cell in the sugar solution contained in the first container is undergoing plasmolysis process. So what is plasmolysis process? This process involves the vacuole and the cytoplasm to shrink and the plasma membrane to move away from the cell wall. But the cell itself doesn't burst because the presence of cell wall. So that's why the mango or the unripe mango is easily due to the plasmolysis process. Next, moving on to the second container which contains the salt solution. So here we can see also that the amount of water is increasing, because that day I remember I only pour 150 milliliter of water inside of this container, and it is only 3/4 than the amount of it is now. Therefore, the second container is also undergo the osmosis process between the unripe mango and the salt solution. Besides that, the plant cell or the unripe mango itself is, it is turgid. Why am I saying so? This is because the vacuole and the cytoplasm is pushing the cell wall. So that's why the cell wall is become turgid. That cause the mango to increasing its actual size. So we can conclude that the salt solution is hypotonic towards the unripe mango. So the third container contains vinegar. This container is also undergoing osmosis, because that day I only put 3 tablespoons of vinegar and the solution doesn't see it clearly, and now it can be seen clearly that the container contains solution. Okay, we take one example. We take the small one. This mango in the third container also undergo plasmolysis. So what is plasmolysis again? Plasmolysis involves the vacuole and the cytoplasm to be shrink, and the plasma membrane move away from the cell wall. But the cell itself doesn't burst because of the presence of cell wall. So that's why it can also be, you see, easily. Same goes to the unripe mango in the first container. So here we can conclude that the third container, which contains vinegar, which shows that the vinegar is also hypertonic towards the unripe mango. So as the conclusion here, we can conclude that the first and the third container, which contain sugar solution and vinegar, is hypertonic, okay, towards the unripe mango, while the second solution, the second container, which contains salt solution, whereby the salt solution is hypotonic towards the unripe mango. So I think that's all from me. See you guys again. Bye.